Hello and welcome to Biochemistry lessons. Today we're going to be talking about proteins with a specific example that everybody likes, which is hair. So hair is made of proteins, made by cells, but it's made of protein. And when you study the structure of proteins, then it's a very good moment for us to talk about hair. So hair is proteins at work. Okay, it's a way that you can exemplify tertiary and quaternary structure working to give your hair the shape it has, the way it is. Hair is made of layers of cells and proteins and stuff. I'm not going to be getting too much details. These are looks of hair. No, they have the cortex, the medulla. No, it's quite complex. If you're into this, you can learn a little bit more about that. But for us, what matters is that the hair will have a certain shape. And the shape of the hair is given by the protein and by the three-dimensional structure of the protein. So basically we are going to be talking here about tertiary structure, about especially hydrogen bonds and G-sulfide bridge. Those two guys are very important for the hair shape. The hydrophobic interactions not much because hair usually is kind of dry, and ionic bonds as well, not very important working here. Okay, so when we wet our hair, the hydrogen bonds in the hair, they loosen up. They change because water influences the hydrogen bonds. So they get kind of different. And you will probably realize that when your hair is wet, it gets a little bit more straight. If you have a curly hair, when it's wet, get a little bit more straight. So this is an effect of you losing the hydrogen bonds with water. So we can place the hair in the shape that we want, and then we can dry it. This is what they do with bobs. They put, they roll around the hair in little bobs, and then they dry it in those funny dryers that you put your head inside. And then when you take off the bobs, the hair is all curly. So they do that for people that have straight hair to have curly hair. What have they done? They have wetted, loosened the hydrogen bonds, put the hair in the position you want, dried it, and remade the hydrogen bonds in the position you want. So the hair now gets curly. The opposite is also done. You wet your hair and you dry it, pressing it with a hot plate. So if you press it with a hot plate, you dry the hair straight, and then when the hair is dry, the hair got straighter. It's no hot plate straightening of the hair. Basically, you are playing with the hydrogen bonds because if you remake it by drying it, then it stays in that place. Unfortunately, both the curling or the straightening using the hydrogen bond idea, wetting and drying, is amazing, kind of quick, kind of fast, very easy, not very aggressive. But if the hair gets wet or even moist, or even if the air in that day is humid, then it changes everything. So you spend 2 hours doing a curly, beautiful hair for a wedding, and then it rains, and you don't even get wet, but the air, when it's moist, it changes, and you lose all the work that you have done. And the same happens with your straightening. You straighten your hair, and then the air is very cold, it's very moist, and you get the hair all curly up again. So to avoid that, there are stuff that you can do. Instead of using the straightening iron or the bobs, there are things that you can do to make things more permanent. So there are some chemicals that break the G-sulfide bonds, and so break the sulfur bonds over there. And you put the hair in the position you want, you put the hair in the shape you want, and then you remake the G-sulfide bridges. This is called the progressive. It's used for doing the perms or permanents or the progressive. So you position and you redo the G-sulfide bonds, and it stays there kind of forever. So these are ways of us playing with hydrogen bonds and G-sulfide bridges or G-sulfide bonds to be changing the shape of our hair. Okay. Some of you are going to be doing a practical later about hair strength and what makes the hair to be strong, but this is for another time. Taking advantage that we're talking about proteins, I want to talk to you about proteomes. You probably heard about genome before, but probably not heard about proteome. So what is a genome? all the genes of an organism, all the genes of a cell, all the genes. And proteome then is all the proteins of an organism, all the proteins of a cell, all the proteins of a tissue. Okay, what about the genome? Genomes is going to be coding for the proteins. So the genes code for protein, and the proteome then is the proteins themselves. So basically one make the other. So shouldn't they be related? Yes, they're related, but not so much. Genome is the potential. If you have a certain genome, that means that you can make some proteins. But a proteome is actually the proteins that you have done, that you made them, not the one that you have the potential to make. Okay? The genome is unique for an entire organism. All the organisms and every cell in the organism have the same genome. So every single cell in your body have the same genome. But the proteome depends on which proteins are made by which cells. So actually you have a proteome for your entire body, but actually every single cell of your body also has its own proteome because it has its own collection of proteins inside of it. Okay? Genomes identical in every cell, proteomes different in different types of cells. Okay, this is the difference between proteomes and genomes. And as we're talking about proteins, remember along the course to go collecting functions of proteins. So far, we have mentioned some of them in the cells classes. We talked about channels and pumps. No, they are membrane proteins. They are proteins that have the function. We talk about hair. We talk about collagen. We talk about hemoglobin, myoglobin, even antibodies. All those are proteins and they have functions. Along the course, go collecting functions of proteins. Someone asks you, what are functions of proteins? Then you can list many functions of proteins around. Okay? I hope you enjoyed and do the quiz. I'll see you in the next class. Bye-bye.